And what we decided to do, with time being limited, I did ask people to submit some questions, and we have got a range of those here. And we've got the bouncy microphone somewhere. Is Robin around? Yeah, he's there. The first question, could I pass the mic to Tony Pincus?
Tony, where are you? Yeah. Right. Tony, you then chuck, this is a nano microphone last week. You then chuck it to the next person after this, and it just works.
Okay. Hi. Okay. Does it work? Does it work? Okay. Well, I'd just like to say thank you for the most amazing conference, one of the best that I've been to in many, many years. Over the panel, which is over the last 3 decades, I've read a large number of published papers on homeopathic studies and even provided remedies for quite a number of these. However, very rarely, if ever, does the author mention or describe how the potencies used in the study were prepared, nor do they venture to discuss, let alone evaluate, different methods of potentization. There is simply a grand assumption that they're all made in the same standardized manner. This elephant in the room is getting larger now that we are discussing the relevance of water and nanoparticles as a transmissible agent for the mechanism of action. As a manufacturer of homeopathic remedies, I've even considered this by designing and building a potentizer that allows a placebo control of the remedy to be produced alongside the active remedy so that the action of the potentization alone can be discerned from the remedy activity. Does the panel consider that more focus needs to be drawn towards this subject of how remedies are made rather than make assumptions that they're all made in the same way?
Yeah. From the point of view of our journal, homeopathy, we do insist that the pharmacopoeia is stated, according to which pharmacopoeia was it made. Beyond that, it does get a bit tricky, I think.
Yeah. I think it's a very valid point generally. If you look in multiple areas of the healthcare sciences, and we look at, say, what the European Food Safety Authority has been doing with nutritional claims of late, they've been rejecting 2-3 decades worth of science because characterization was not sufficient. So if you look at, say, the rejection of hundreds of probiotic claims, most of that is down to poor characterization. Very simple solution that you characterize it in detail once you publish it, and then in the methodology you refer back to the published study and everyone knows exactly how it's made. So these are just points that can be dealt with through good science.
I'm not talking about remedies. You see, it's only my organism can talk about this. I would like to talk about the preparations for studies in vitro, so to say, action on enzymes, on cells, and so on. Here, there should be some minimal demands for preparation of such, for making such preparations. What I mean by minimal demands, that is just no agitation, no less than some threshold limit. So below this threshold limit, you simply just make dilution without agitation, it will not work. If you make this agitation, it will work, but it may work in very different way, depending upon factors which we usually do not pay attention to. I told about the role of electromagnetic environment in these preparations and so on. And this electromagnetic environment, especially natural, it changes continuously. We have geomagnetic storms. We have solar eclipses and rising moon and going moon which is going down. All these factors may affect the result, the outcome. I'm talking now only about in vitro studies. As regards in vivo studies, it is probably on one hand more complicated and on another hand more simple. because the organism may be very sensitive to even minor activity of these homeopathic remedies, or very insensitive even to just to strong remedies. So the science, to my mind, is the just starting position to take into consideration all these factors of environment and to study how to work with them. I would like to remind about the gardening. Yes, many people who are making gardening, they know that it depends very much, the result will depend very much on moon. You see, for academic science, it is considered to be just complete nonsense. But those people who are practitioners, they know very well that, for example, radish could not be seeded at this particular time and only 3 days later. So it seems to be like miasms, but it really works.
Thank you, Vladimir. Thanks. Dr. Wayne Jonas, you have a question? We got the...
Please stick your hand up so Jack the mic.
I think you also have such an experience that not experiments do not reproduce.
The other way,
Perfectly.
All right, so we've talked a lot about structures, molecular structures, propagation into water, how that might get and then perpetuated through electromagnetic signals, etc. One subject that keeps coming up is the idea of quantum and quantum interaction. And we know that quantum interaction has an effect that is not just electromagnetic. Okay, there's something else going on. We also know that it interacts with mind. I think, as Professor Joseph, and you're the only one that explicitly mentioned the mind and the matter, that there might be some type of quantum entanglement. And although I know we want to be very conservative and we're all uncomfortable when we get outside of the molecular discussion, that I wonder if the panel would talk a little bit about the interaction of the mind and of quantum non-locality in terms of the delivery, especially around semiotics, and how that might interact with some of the other mechanisms that have been discussed today.
Professor Joseph?
Well, I think entanglement is a very general mechanism, and you can, I guess it's two systems coming into terms with each other, and to some extent you can talk about it in terms of quantum mechanics, to some extent in semiotics, but I believe a more wider approach is necessary. But the concept of entanglement, in fact, well, people in quantum mechanics are beginning to acknowledge that entanglement is important and a bit mysterious, and a wider significance than the standard experiment in entanglement, where you have one system which disintegrates into two, and these two get entangled, and it's being recognized that that's an oversimplification of what can happen.
Thank you. Dr. Tarako Yui. You got the cube? Thank you.
Okay. Well, I asked the Professor Montagnier. I have been very impressed by your study and have been convinced that medicine should be changed through your study. I had understand about your study as below, and would appreciate if you could confirm that my understanding is correct. DNA of HIV was diluted to 7x to 13x, sarcastic or not. Then the water have interpreted the electromagnetics signal into PC using the electromagnetic signal nano EMS. When the electric magnetics signal was fed into water, which did not have the DNA of HIV at all. The DNA of HIV was restored. What I would like you to confirm is as is showing below. I believe that gene of HIV is RNA, not DNA. You are created DNA of HIV by reserved transcriptase and used it as an experimental material. Is this correct understanding?
That's a lecture, not a question.
Or not? Also.
This is a Russian method. This is a Russian
To answer your last question, HIV DNA is found in nature. This is the usual cycle of this kind of virus. It is from RNA, but inside the cell, it makes DNA, which integrates inside the genome DNA. So, what is your question is? Shall we reproduce what nature does? Well, this is the main problem of HIV infection, actually. HIV infection state persists in the body because of this translation from RNA to DNA. So this is not a problem created by us, it's created by nature. So we have to solve it. To my more general comment, I didn't get all your question, but my more general comment is that we are now in a stage in which we can really make studies on some pure material, because we can make dilution. We know that in some dilution there is nothing left from the original DNA, so we can study pure structure, water structure, which may differ on different dilution. And as we have seen, there are holes, in some dilution works, some do not work. I think this is a very important question. Does it compare to the therapeutic effect? Yeah. Is the same in some dilution which are negative or which are positive? This is some important question we can solve now with some pure material which could not do before in other systems. This is my feeling, my dream.
Thank you very much. Just one or two more. Enid Seagal? Enid Seagal? Okay.
And because I submitted my question, I didn't bring it with me, but I will summarize it. I looked in the dictionary, and it says that a scientist is questing for knowledge and trying to solve the problems that come up in nature and health and whatever. And yet, I spend a lot of my time dealing with the problems created by the opposition to homeopathy at the hospital in London. And so I wondered how we can persuade these people who oppose homeopathy to actually look into it, because I've had conversations with them and they know nothing about it at all. And so I'm wondering whether this conference is the big breakthrough that we've been waiting for.
Well, I hope so. You know, it's been a mammoth task, and we've given our best shot. And I might just add that I'd like this to be the start of something for the future, not just a one-off. I'm actually considering maybe that we could form a foundation where everybody's included, all the researchers, and more governments are involved, such as Israel and India. And maybe next year we can take the water forward, you know, the water topic and homeopathy. Maybe we'll do it in Jerusalem or Delhi or somewhere in New York. We've had all these offers coming in. But I think we'd like this to be the beginning, not just a one-off occasion. Thank you.
Could I comment on that? Well, you have to look into the reasons why people don't look at things, and the common explanation is that it's impossible. For example, another example is that telepathy is considered impossible, and a well-known physicist has said it's so unlikely that it can be true, it's not worth my time looking into the evidence. So I would say the appropriate response is to ask them why they think it's impossible and then shoot down their argument.
Thank you.
Well, I'm more pessimistic. I mean, based on the ideas of Thomas Kuhn, most probably we will not convince them. We must wait until this generation will disappear. And then
Just one or two more, then we have to wrap up. Tony Scott Morley. Yeah.
Some years ago, I did an experiment where I transferred information from a sealed glass ampule of a homeopathic medicine into a sealed ampule of plain water. The pH changed by several orders of magnitude from 7.3 to 7.7. The electrical conductivity was in agreement with what we've heard today. Now, I don't understand how this could be based on nanoparticles, since it was only an electronic transfer. Could you please comment?
Well, everybody's looking at me, so I should answer. Sir, I can be rude and tell you I should repeat your experiment. But I think I'm willing to work with you, but I see no explanation for how that happens. And I despite the discussions we've had today, it is my firm belief that all action depends on the presence of a material system, and the material which is present at these trace amounts seems to have an effect, and it is the material which is responsible for this. Now, if I can extend this for a moment, which I have no proof of, perhaps the presence of nanoparticles in one system, the vial, the sealed vial, had some electrodynamic or quantum chromodynamic effects which might have affected the other one. But the presence of those nanoparticles in one system was essential to cause the effect. So that's the best explanation I can give without doing further explanation.
Did you at any point put another vial in without any homeopathic substance in it?
Yes.
And you didn't get the effect?
I didn't get the same effect. I also noted that there was a difference in the change in pH depending on whether the substance was succussed or merely stirred.
Right.
And there was a clear difference. The two lines ran parallel, but the succussed was a stronger effect. But going back on that last reply, perhaps we should be cognizant of the work of Christian Endler in Graz, Austria, who did a very interesting experiment whereby he suspended a sealed ampule beyond the Avogadro number into, it was an ampule of thyroxine suspended into a tank of tadpoles and compared with the control group. And the tadpoles in the experimental group matured into frogs at roughly twice the rate of the control group, which again suggests we're dealing with some sort of electromagnetic information field.
I just would like to add that your experiments are not unique.
No.
Yes, we also performed such experiments and we got same results when sealed ampoule is filled with, by the way, homeopathic dilution of fullerenes. And these fullerenes, we even published such a paper, not about ampoules because I'm not. So it really works. Why it works? When we are talking about nanoparticles, I think there is a division between your experiments with the nanoparticles which you can locate and nano-associates which practically consist mostly of water. And the Italian scientists, they made it just on pure water without any initial nanoparticles and so on. So, and they got this water which was not evaporating and things like this. So this cannot be explained on material transfer. It can be explained only using the idea of dynamic state of the system. And when the system is in dynamic state, it is the source of very probably low intensity, extremely low intensity, but of radiation. It may be just electromagnetic radiation and all kinds of radiation, of course, which can penetrate any glass and things like this. So that is just the form of interaction at a distance. And for us, we consider it so difficult, that interaction at a distance, but believe that at the end of the 19th century, nobody could think that somebody talking in Britain will be heard in America. And so the voice could be transferred to another continent. So we have here also in our, that's interesting, that in our biology, we do not believe that we can talk to the living systems can talk to each other without direct transfer of material letters.
Brian, did you want to add something, Brian?
Well, I sort of agree with what's been said, and if you have a theory that systems can't talk to each other without material transfer, how is it that I'm talking to all of you, where it's just the energy and information being transmitted?
Thank you very much.
Yeah, there's a very interesting experiment that was reported at the water conference by a guy named Serge Kernbach. And what Serge did was to look at water, and he uses a very sensitive method called impedance spectroscopy to look at the physical characteristics of the water. And he has careful controls, and he has people focusing their intent on the water from distances up to 10,000 miles away. And if you see the result of a person focusing their intent before and after, the result is dramatic. It's really an interesting experiment because it's a physical way of determining that you can have information transmission from a great distance, and it really has an effect on the water, as many of us had surmised, but this is a rather direct observation of such.
Thank you. I think just, just two more questions, and our American ambassador for homeopathy does a heck of a lot, Dana Ullman.
Oh, wow, that's great. It's good to stand up, first of all. The worldwide marketplace for homeopathy is probably around $2 billion, but if you followed the marketplace for this broad field of nanopharmacology, in a couple of years it's expected to be $100 billion. And ultimately, if we think about it, homeopathy is the original nanopharmacology. And I encourage us all to use that term, homeopathy, as the original nanopharmacology, because it begins to
provide us with coattails that will take us into the future. In the story of whether it's nanoparticles or electromagnetic, like the wave particle theory, it's both. It's clearly both. Entomologists have found that pheromones act on a single molecule. And so whether it be just a single molecule that's able to penetrate the blood-brain barrier and create an immunological reaction, that I'm sure happens, as does the electromagnetic fields. But the additional comment I wish to make, there's been some interesting research on plants and arsenicum. And they find that the making of a homeopathic medicine isn't as active in the first day and actually increases over time. So part of our research also has to include when the medicine was made. And I can't help but wonder, but if the nature study of Benveniste, when they just made the medicine, if maybe that was part of the problem. They needed to make the medicine a couple weeks, couple months ago or something like that. And that has to be in some of the literature too.
Thank you very much.
Professor Pollock gave us actually two avenues, by an electromagnetic avenue and a physical material avenue. And I want him to ask you a question about the more simple-minded material thing. So when we think that substances will be coated with easy water, and you showed us this amazing experiment by this Italian group where they made this easy water actually visible at room temperature as a kind of crystals. Is it thinkable that these easy water produces like a mold around a protein which would act as a receptor? And then through the shaking, this mold is broken up and into nanoparticles. And can then these nanoparticles of easy water create more easy water around them? Is that thinkable?
Absolutely, yeah. I mean, I think that this could easily occur during succussion, where first of all, your first point as a mold, actually so, and I was going to comment on the nanoparticles that you were discussing, and they too are likely to have easy water coating the surface. And during succussion, it seems natural that as you shake, as you agitate, these structures will come apart, and they can act as nuclei for additional growth of easy water. And if I were to speculate, I'd say that's what happens during succussion, is that this easy water is broken up into a lot of little easy waters, and that they can carry the information onto successive succussions. So if that answers your question.
Yeah, I went to Professor Montagnier because you showed that you could materialize a DNA molecule out of nowhere in water through just sending some information over the internet. And if we can think that obviously some electromagnetic field can also change the structure of water into a kind of mold, then these rudimentary elements could then assemble themselves inside this mold. Is that maybe an explanation?
Yes, one question we haven't solved yet. In the size of that DNA we can produce today, it seems that they are small fragments so far. I won't say today that we can reconstitute microorganism DNA, bacteria, the complete bacterial DNA that way. But this DNA, even though it is in small fragments, carry genetic information and can therefore interact with the genomic DNA in some ways. So this is an open question, but not yet solved. But I think the hope coming out from this meeting today is that now we have some pure material. We have also a unifying concept, which is water nanostructure. Nobody discussed that anymore. At least in our meeting. So this is an important move, which make it possible to make further study, by released, biophysical tools, mind effect, and so on. We can try to solve this, but it will take time, research, and money.
Right. Now you comment on that? Yeah, I'd just like to make a connected remark, but I see the origin of life question in a different way from the conventional. If you're going to start off with chemistry, then it's difficult to get life from it. Whereas if there's something at a deeper level, which, as it were, has had experience in putting things together in complicated ways, then you may be able to build up step by step, maybe even building up life, the universe, and everything from a simple starting point. I think that's a more plausibly explains what goes on in the origin of life. And life learns to make what kind of universe is best to support a more advanced form of life. Incidentally, I have some YouTube videos on this. If you search me, you can find out more about these ideas already.
I just want to make one remark now that we're, I think, about to close. I just want to say that this meeting has been spectacularly organized, having organized many meetings in the past. I just ask one favor. He has to decide whether his name is Aaron or Ken.
So we conclude the meeting. Keep in touch. We're looking at next year. And thank you for coming along. I know many people have come from out of the UK, and I wish you a safe journey back. Thank you so much. Thank you.